And this is Chapter 17, Part B. So this is going to be specific response via the T lymphocytes. Last one were the B lymphocytes. Okay, now, as much as this is going to emphasize the T lymphocyte response, this is kind of a review slide of the role that the antibodies play in the specific immune response. We talked in kind of vague generalities that when antibody binds, it can cause cascades that lead to the destruction of a pathogen. This goes into a little bit more specifics. So going clockwise, let's might as well do this. Activation of complement. Complement is something that I'm briefly going to explain. It's a very complicated cascade, but when antibodies bind to antigens on the surface of a this case, a pathogenic bacteria, it in and of itself is not capable of killing. So it needs to bind and then to elicit other mechanisms and other cells to do the killing. But what they serve the purpose is to identify those cells that need to be destroyed, and they should only bind to pathogens. They should never bind to your normal body cells. So complement is a cascade that can be activated when antibodies bind to the surface of a pathogen, and then complement starts a cascade which will cause the death or lysis of the pathogen. Now over here, inflammation, we've gone through that before. That's a non-specific response, but antibodies can also amplify the inflammatory response. Then we have what is called antibody-dependent cell-mediated cytotoxicity. Very long name. What it essentially means is that when an antibody, that little Y-shaped molecule right there, binds to, let's say, a pathogen, as well as activating complement, it can also draw to that specific site other types of cells, such as phagocytic cells, that can then either engulf or release cytokines that are signals that will start a cascade that will destroy the pathogen. Over here, neutralization. Sometimes you have free viruses or you have toxins, and they can be neutralized. This is a fancy name for saying that antibodies can bind to a free virus, antibodies can bind to a toxin, and then that blocks their ability to bind to a target cell. So viruses that are neutralized by antibodies cannot bind to a cell to infect it. Toxins cannot bind to cells to poison it. Instead, these antibodies that are surrounding a free virus or a toxin will get swept out of the body via usually urine. It'll get filtered by the kidney, urinated out of the body, so the virus no longer can attack you, the toxin can no longer poison you. Now, opsonization, it comes from an interesting Latin word, which literally means to make into a tasty bit. And in a way, it's appropriate because when you have antibody that is bound to a pathogen, yes, you can absolutely positively have phagocytosis to eliminate it, and this is what it's showing you as a phagocyte. But when antibody is bound to the pathogen, it will phagocytize it more readily, more easily, more ferociously, because now it's made into a tasty bit rather than just a bit. So that's opsonization. And then last but not least, agglutination. When antibody binds to bacteria, besides eliciting opsonization or complement activation or a cell that can phagocytize and kill it more rapidly. It can also cause the bacteria to agglutinate. So it's not just binding to one, like right over here, it's binding to one. It's binding and forming crosslinks between adjacent bacteria. And that would mean that the bacteria itself could agglutinate, which would allow the kidney to eject it from the body more easily in the form of urine. It also could mean that the phagocyte would engulf a number of pathogens, not just one. It could, if you have a fair amount of agglutination, then you could have a phagocyte engulf 10 or 20 at one pass. So these are all of the different ways that antibodies target, and antibodies will elicit other cells and other mechanisms to help destroy the pathogen as well. So that's what that slide is. Almost like a end to the other lecture. We had talked about a memory response, and this is just showing you a diagram, a graph, that proves it. So what they've done is they've taken, and this is usually not people, they do these experiments in rats and mice, but then they also have observed the same thing in humans. So here is the amount of antibody, and usually the memory antibody is IgG, and this is the time after the initial infection and after reinfection. So here's an antigen, and here's the response. So here you have IgM and then IgG. Note that both of them are about the same amount. And then the test animal recovers, and then they are re-exposed. And now they're watching for the memory response. Now notice that after the initial infection, you get kind of the height around probably day 7 to day 14. Now you're exposed, and now it's, let's say that this is around 25, and then within probably 3 or 4 days, you really start to have an upward surge of IgG, not so much IgM, but IgG, so that you could clear that infection so rapidly and so vigorously that the person doesn't get sick. So that's memory response. Okay, what happened to that slide? This is a slide that is a little off. This goes into the different types of T cells, and I'm having a hard time bringing this little sucker down. I'm not going to bring the sucker down. So we're going to go back, and I'm going to tell you what this says right here. This is called a T. These are types of lymphocytes that get involved with specific responses. And this is a T helper lymphocyte, also called a TH lymphocyte. And because of its receptor and because it's singled out by the HIV virus, it's also called a CD4 cell. And we had talked about that when we talked, oh, I'm sorry, we didn't talk about it when we talked about AIDS. This is, actually we did talk about HIV and AIDS. So we talked about the CD4 receptor and how when a person is being monitored for their HIV titer or their AIDS response, that they talk about the person's CD4 cells or T helper cells, because that's the only cell type that's infected by the virus. Okay, so that is really like the first cell that is activated when you elicit this specific response. So it says activate cells related to cell-mediated immunity, activates B cells to produce antibody. Other types of T cells. Another type besides T helper is T cytotoxic cell, also called CD8 because its receptor is slightly different, and the nickname is TC, standing for T cytotoxic. Another nickname is killer. So cytotoxic T cells or CD8 cells or TC cells can also be called T killer cells. Why? Because they bind, and when they bind to the pathogenic cell, the target cell, they cause cell-to-cell contact killing. And we'll go into the mechanism in a second, but essentially they act as hypodermic needles. They have a certain amount of poisons, and then they will bind to the cell and shoot in a little bit of the poison like a hypodermic needle, and then disengage from the cell. The cell dies, and it can go on to bind to another pathogen, inject a little bit more poison, and then go on and on and on. And they can kill about 10 to 20 cells before they run out of their poison in their proverbial syringe. Then we have suppressor T cells or TS cells, and they can turn off the immune response when the antigen or the pathogen is no longer present. And that way, you don't run on high gear all the time. You run on high gear, you get rid of the pathogen with its pathogenic antigens, and then you don't need those any longer. You've resolved the issue. So T suppressor cells will then turn off that immune system. You will have memory, but you won't have the amount of T cytotoxic cells and antibodies because you don't need them anymore. There are two other cell types that I want to talk about. The natural killer lymphocyte, it's called an NK, natural killer cell. It's not a T cell per se, but it's very closely related and does cell-to-cell contact killing very similar to the T cytotoxic cells. And the other cell that I want to mention, of course, now there it is. It's called a dendritic cell because it kind of looks like a dendrite to people, but you have them scattered throughout your body. They're there in the epidermis as well. And they are another type of cell that can capture antigens from pathogens and present them to T helper cells. All right, so let me go back. All right, to this slide. T helper cells communicate. How? They make signal molecules, and the signal molecules then can diffuse to other types of lymphocytes and activate them, stimulate them, get them to grow, get them to multiply, get them to arm themselves, get them to release antibodies. The two most famous are interleukin 2 and interleukin 1. So they're called cytokines, meaning they're cell signals, and these are the most famous. Now, interleukin 2 is going to be released by a presenting cell called a macrophage, meaning large cell. It also does phagocytosis, but it does probably 12 to 24 hours later. So if your neutrophils fail, they can't phagocytize enough, then when the macrophage comes 12, 24 hours later and still finds an area is infected, it will do phagocytosis, but its job is to activate the lymphocytes, to specifically activate the T helper lymphocyte to start the specific response in order to save the person's life. So interleukin 1 is secreted by dendritic cells that present antigens or macrophages that present antigens, again, 12 to 24 hours later, and because you have not yet cleared that pathogen, you need more help. So they are going to present it to the T helper cells, and interleukin 1 is one of the stimulatory chemicals that are made by macrophages and dendritic cells to stimulate T helper cells. Interleukin 2 is a signal made by the T helper lymphocyte. Now look at what it can activate besides T helpers. B cells, B lymphocytes that produce antibodies, T cytotoxic cells, killer cells, and natural killer cells. So all of these are going to get amplified, and you're raising up a great army to go out and do all sorts of killing because you don't have a fourth line. You're either going to have the third line of defense succeed or that person can die. Okay, so cell-mediated immunity is different from humoral immunity. Humoral immunity is B cells making antibody. Cell-mediated immunity is all about T helper, T cytotoxic, natural killer cells, and memory cells. So specialized lymphocytes, mostly T lymphocytes or T cells responding to pathogenic antigens. Now they get presented by macrophages or dendritic cells. They are called T cells because they mature in the thymus gland. Now, the thymus gland lies on top of your heart. And this thymus gland is quite large in infants and young children, and then gets to be much smaller. It starts off probably like a very small walnut, and right about now my thymus is the size of a shriveled pea. But the thymus is where the T cells mature, and then once the maturation has occurred, you store them in lymph nodes. T cells differentiate into effector T cells when stimulated by pathogenic antigens, such as T cytotoxic cells. Some effector T cells become memory cells. And there's our dendritic cell, and this is a macrophage. So this has an awful lot of detail. I'm going to try to just highlight some things. The macrophage arrives late. If it still sees that you have the infection, it will engulf the pathogen and then strip it of its antigens, these non-self molecules, and stick them on the surface presented in the lymph nodes to a T helper, releases interleukin-1, and then the T helper gets activated, releases interleukin-2. Now watch what interleukin-2 does. It's going to stimulate more T helpers. It's going to stimulate the maturation and growth and multiplication of T cytotoxic cells. It's also going to stimulate B cells to produce antibody. And then you have mounted a really good army to go out to the infected site, and on many flanks simultaneously antibodies arrive, natural killers arrive, T cytotoxic cells arrive, and they are going to decimate the pathogens, and you will develop memory as a result. And that's all I really want you to know. So this is showing you how it operates as far as the cytotoxic or natural killer cells. The only thing I don't like about this is it doesn't really show the syringe-like apparatus. But think about a syringe and think about it being loaded with these toxins, poisons, some of which are called porphyrin, and other types of lethal chemicals. And so they do make cell-to-cell contact. Here is maybe a virally infected cell that you want to get rid of before it actually matures the virus. And it binds to the antigen and then injects the poisons, and that is not going to hurt the T cytotoxic cell and the natural killer cell. It's just going to disengage and go off to bind to another and another and another, and then the poisons and the porphyrins do their deadly job, and the cell dies by lysis. And that is it. Oh, I'm sorry, almost it. I know you're just dying to see what a macrophage looks like and dying to see what a natural killer cell looks like. Okie dokie. So this is a macrophage, and then here are some activated macrophages. The natural killer cell looks like a lymphocyte. A macrophage is a very large phagocytic cell, and there we go, a macrophage, an activated macrophage, and that does have to happen first before it presents it to the T helper cell, which then activates a whole army of effector cells in order to eliminate the invader for good. And that's it. So that's an overview of the immune system, both the kind of the natural and versus acquired and innate versus specific immune response. And it's a brilliant system because you have a 24/7 type of protection with the inflammation of phagocytosis. And if you have very persistent pathogen that is trying to gain a better foothold and is not able to be stopped by the inflammation in phagocytosis, then you can activate an army of lymphocytes that will go out and very vigorously and robustly decimate that persistent pathogen and get specific memory because of that. And that's it. So I will see you for the next week. And the quiz is going to be based on these 3 chapters. What I've gone over, not what I've not gone over.